Up to now, our lecture series is focused on the visual system and the auditory system, and to a lesser extent, the chemosensory systems of taste and smell. These systems are really about making perceptual analyses and decisions about what is out there in the world. That is, to identify what objects are, and also to work out where they are and maybe in which direction they're moving, and so on. In today's lecture, we're going to talk about two other sensory domains or sensory dimensions, which we call the bodily senses. These tell us how we personally are situated within the world, as well as how we are interacting with the world. The two sensory systems we'll discuss today are the vestibular system and the somatosensory system. The vestibular system provides us with information about the movement of our head and its orientation, while the somatosensory system gives us information about touch, temperature, pain, and body posture. The vestibular system is involved in analysing our head's movement through space. It also gives us a sense of visual stability. So when we tilt our head, our eyes will actually right themselves to remain as horizontal as possible. And it uses our vestibular system to do this. We know the vestibular system is important because when it goes wrong, we experience problems like spatial disorientation or dizziness, vertigo, imbalance, blurry vision, and illusory self-motion. These issues can arise from several causes, such as ingesting certain chemicals like alcohol, inner ear infections, and even some viruses. It's also caused by other things like adaptation to movement, such as when we spin around for a long time, we get dizzy. This is caused by adaptation within our vestibular system. There are several aspects of vestibular system functioning. The first is angular acceleration, which enables us to perceive changes in the direction of our head's movement or orientation. Angular acceleration is mediated by what are known as the semicircular canals. And these are located within each ear, or just above the cochlea. We're also sensitive to linear acceleration, which differs from angular acceleration as it involves changes in the direction of our head without any changes in its orientation. The vestibular system outputs two dimensions: the amplitude of our head's movement and the direction of this movement. Angular motion involves changes in our head's orientation across three dimensions: pitch, yaw, and roll. Pitch refers to nodding up and down. Yaw refers to shaking the head from side to side, and roll refers to tilting the head side to side. Linear acceleration, on the other hand, involves changes in direction without changing head orientation, such as when we move backward or forward, side to side, or up and down. The main thing to remember is that angular acceleration involves changes in head orientation, whilst linear acceleration does not. The vestibular system is not only sensitive to changes in direction, but also to the orientation of our head due to the force of gravity. The vestibular organ converts head direction into neural representations, with 2 sets of organs associated with each ear. Each ear has 3 semicircular canals, each oriented in different orthogonal planes, allowing us to detect pitch, yaw, and roll. These canals are fluid-filled chambers with a region called the ampulla, where transduction occurs. Inside each ampulla are hair cells within the fluid, similar to the cochlea. This concludes the initial part of our discussion on the vestibular system. Next, we'll delve into the specifics of how the hair cells within the semicircular canals function to detect head movement and orientation. So, here's the fluid-filled chamber in the ampulla, and here are the cilia within each ampulla. Around each cilium is a pouch called the capsula. Now, when you move your head in a certain orientation, a movement is set up in the fluid-filled semicircular canals. The fluid begins to move due to inertia. As it moves, it pushes the capsula to one side, displacing the hair cells as well. These hair cells release neurotransmitters upon their displacement. Depending on the direction of the push, we get excitatory or inhibitory responses. What I'm showing you here is a hair cell bundle known as a stereocilia. On the left, we have the shortest hair cells, and on the right, we have the longer hair cells. When the hair cells are pushed in the direction of the longer hair cell, we get excitation. If pushed towards the shorter hair cell, we get inhibition. Now, let's discuss the relationship between the movement of our head and the response of the stereocilia. Focusing on one of the semicircular canals sensitive to angular motion, specifically yaw. The yaw motion, remember, is the motion of our head turning left or right. Imagine turning your head to the right, that is, clockwise. The effect of turning your head clockwise on the semicircular canals is that the fluid will initially move in the opposite direction, that is, counterclockwise, in one of the semicircular canals, that is, the semicircular canal linked to yaw. In the left of these canals, this counterclockwise fluid movement pushes against the capsula, displacing the stereocilia towards the shorter hair cells, causing an inhibitory response. In the right canal, the fluid movement pushes towards the longer hair cells, causing an excitatory response. This complementary activity of inhibition in one side and excitation in the other informs the brain that the head has moved in a clockwise orientation. The same principle applies when moving the head in the opposite direction, generating complementary but opposite responses in the canals. This mechanism helps us understand our head's orientation and its movement. Now, let's shift our focus from angular to linear acceleration. Besides the fluid-filled semicircular canals, we have other modules called otolith organs, which are sensitive to linear acceleration. These organs, like the semicircular canals, have stereocilia, but are covered with a layer of crystals known as otoconia or otoconia. These crystals amplify the inertia signal, enhancing our sensitivity to linear movement. For instance, if you move a plate of jelly back and forth, the jelly will initially move in the opposite direction due to inertia. Adding a layer of the crystals increases the inertial signal, making the movement more pronounced. The otoconia function similarly, providing information about linear acceleration and amplifying this. Moving on, let's discuss the somatosensory system, which comprises our sense of touch, temperature, pain, kinesthetic sense, or the awareness of body parts in relation to each other, as well as our proprioceptive sense, that is, our internal sensations like hunger and our heartbeats. Our sense of touch, or tactile system, is a response to variations in pressure on the surface of the skin, mediated by 4 types of receptors: Meissner corpuscles, Merkel cells, Ruffini endings, and the Pacinian corpuscles. These receptors differ in their location within the skin as well as their function. Some respond to light touch and have small receptive fields, while others respond to deep pressure with larger receptive fields. Receptive fields vary in their size. Merkel cells and Meissner corpuscles have small receptive fields and are in the outer skin layers, providing fine detail. Ruffini endings of Pacinian corpuscles have relatively larger receptive fields and are located deeper in the skin and respond to broader pressure changes. Each receptor type also has different temporal responses, or responses that occur as a function of time, processing pressure changes at various speeds or temporal frequencies. Merkel's and Ruffini endings respond to what we might call slow changes in pressure or low temporal frequencies. What do I mean by slow changes or low temporal frequencies? Well, a slow change occurs when you're interacting with the world, such as when you hold a large object, such as this remote, and move it across your hand. Move your hand across it. You can feel the object itself, the shape of the object. As you move your hand across the object, you can feel its overall shape. This produces a fairly slow, sustained temporal response, which is picked up by the Merkels and the Ruffini endings. By contrast, Meissner's and Pacinian corpuscles respond to much faster changes in pressure, that is, high temporal frequencies. When do we encounter fast changes in pressure? One common way is when you rub your hand across the surface of something. If you feel something rough, for example, you may detect very fast, small changes in pressure as you move your finger across the surface. This is a consequence of responses in the Meissner and the Pacinian corpuscles. Another example is when you experience vibrations, such as when your iPhone vibrates or when you feel the vibration of your own voice in your throat or in your chest. These sensations are also detected by Meissner's and Pacinian corpuscles. Moving on to another sensory system, we have the kinesthetic system, which is the sense of our own body's orientation and movement. When you move your arm back and forth, you can sense where it is and how it's moving, based on the specific receptors. Here we see muscle fibers and different types of fibers like alpha and gamma. Here we see muscle fibers and different types of fibers like alpha and gamma fibers, which are sensitive to our own movement. When you move your arm, you sense that movement through these fibers, and you can distinguish between your arm being extended or flexed based on type 1 and type 2 sensory fibers. Now, let's discuss thermoception, our ability to sense temperature. We have different types of fibers that mediate this sensation: warm fibers and cold fibers. Cold fibers are most sensitive to about 25 degrees Celsius. They have relatively broad bandwidths, meaning they can also respond to colder and warmer temperatures. Warm fibers are most sensitive to about 45 degrees Celsius. These also have relatively broad bandwidths. When you make a perceptual judgment about temperature, you're actually evaluating the relative responses of the cold and the warm fibres. We've seen this before, this relative response in our perceptual judgments of things like colour. Remember, our cones have broad bandwidths and they're overlapping. So when we perceive the colour of something, what we're doing is evaluating the relative response of the different cone types. Similarly, when we make a perceptual judgment about the temperature of something, we're making a relative judgment or a judgment about the relative response of the cold and the warm fibres. Another subsystem is nociception, or the pain system. We have two types of fibres sensitive to pain, A delta and C fibres. A delta fibres are highly myelinated, allowing signals to travel much faster, resulting in immediate sharp pain. This is followed by a more sustained throbbing pain from the C fibres. All these fibres, whether they're thermal receptors or nociceptors or tactile receptors, travel from the body through the nerves to the spinal cord and then to the thalamus. The thalamus acts as a sensory relay station, sending signals to the primary somatosensory cortex in the brain. The primary somatosensory cortex, or S1, is responsible for processing these signals and is located in both hemispheres of the brain. Information from the right side of the body is represented in the left somatosensory cortex. Information from the left side of the body is represented in the right somatosensory cortex. This is known as a contralateral representation of the body. Where else did we see a contralateral representation? We saw it in the retina, right? The right visual field is represented in the left visual cortex. The left visual field is represented in the right visual cortex. What I'd like you to notice is that in the primary somatosensory cortex, we find that the neurons there are sensitive to different parts of the body. Importantly, we find that there's a continuous representation of the body across the surface of the cortex. We call this a somatopic map, meaning that adjacent neurons in the cortex encode adjacent regions on the body or the surface of the body. This is analogous, or this processing strategy is analogous to other sensory dimensions as well, other senses, including the visual system, which has a retinotopic map, and the auditory system, which has a tonotopic map. Remember, the retinotopic map means that adjacent regions of the visual cortex are sensitive to adjacent regions of the retina. In the case of the auditory system, we have the tonotopic map, whereby adjacent regions of cortex encode adjacent frequencies and therefore adjacent regions of the cochlea. Similarly, we have a somatopic map, meaning that adjacent regions of the cortex, of the somatosensory cortex, are sensitive to adjacent regions of the body. Now, importantly, yes, there is a somatopic map, but it's a distorted map. It's a disproportionate map. Some regions of the body are disproportionately encoded by the brain. That is to say, there's far more regions of cortex dedicated to certain body parts than there are to certain other body parts, in a way that's disproportionate to the actual size of the body. This is represented here in the image on the right. This is an artist's rendition of the amount of the image of a person whereby the size of their body parts reflect the amount of cortex dedicated to each body part. You can see that there's far more cortex devoted to things like the hands and the fingers, as well as the mouth, the tongue and the lips, than there are to other body parts. Why might there be more brain regions devoted to the hands and the mouth? Well, we use our hands for various activities, such as writing and typing and driving and exploring the world, often using them to manipulate tools and objects, and even each other. Infants also explore the world through their hands and their mouths. Similarly, the mouth is heavily involved in speech production, requiring precise and nuanced movements to produce meaningful sounds. In summary, the disproportionate variation in somatosensory cortex dedicated to different regions of the body reflects the importance of different body parts in not only sensory processing, but our interaction with the world. That is, they have an articulatory and communicative benefit, as well as subserving things like tool use. The phantom limb phenomenon is a fascinating and sometimes distressing condition experienced by individuals who have a limb amputated. Despite the absence of a physical limb, individuals often report sensations including pain, as if the limb was still actually present. This phenomenon provides a compelling insight into the brain's role in sensation and perception. The sensation of a phantom limb arises because the brain retains a map of the body in the somatosensory cortex. When a limb is amputated, the corresponding area in the cortex remains active and can generate sensations spontaneously. These can range from tingling and warmth to severe pain, often described as burning or stabbing pain. Research suggests that phantom limb pain may be related to the brain's attempt to reorganize itself following the loss of a sensory input from the amputated limb. This seems like a maladaptive reorganization, as it causes neurons to become hyperactive or misfire, leading to the sensation of pain. There are treatments, however. One of the more effective treatments is mirror therapy, where patients actually use a mirror to create the illusion of the missing limb actually being present on the body. This has been shown to help reduce phantom limb pain by helping the brain recalibrate its perception of the body.